So here's a question that has haunted every neuroscientist, every AI researcher, and every undergraduate who has ever started at a textbook at 2 in the morning wondering why their brain wouldn't cooperate. How does your brain know which neurons screwed up? No, seriously. You have roughly 86 billion neurons. 86 billion, okay? That's more neurons than there are stars in the Milky Way. We love that comparison because it makes us feel cosmically important, so we're keeping it. Every time we learn something, catching a ball, recognizing a friend's face in a crowd, figuring out that stove is hot and you should probably stop touching it, your brain has to figure out which specific neurons out of those 86 billion were responsible for the mistake. Which ones need adjusting? Hey, you specifically, you did this wrong. And here's exactly how much, and here is a direction you need to correct. In computer science, this problem has a name. It's called the credit assignment problem. And it is arguably the single most important problem in all of learning, biological or artificial. And for decades, the answer in AI has been a beautiful, elegant algorithm called backpropagation, or backprop, if you want to sound casual at parties. Backpropagation is the engine under the hood of virtually every AI system you have ever heard of. ChatGPT backprop, self-driving cars backprop. That algorithm that keeps recommending you videos of baby goats in pajamas, backprop. Backprop is the thing that made deep learning deep. And here's the twist. For 40 years, neuroscientists have said, essentially, beautiful algorithm, love it. The brain cannot do that. The brain cannot do backprop. It is biologically implausible. The wiring is wrong. The timing is wrong. It's like saying your grandmother could run a marathon. Lovely idea, but the hardware does not support it. Until 3 weeks ago. Because in February 2026, a team at the Massachusetts Institute of Technology at MIT, led by Mark Harnett, published a paper in Nature that changes the game. They found the first biological evidence that your cortex, your actual physical meat-based brain, solves the credit assignment problem using vectorized instructive signals in dendrites. Translation: your brain is doing something shockingly similar to backpropagation. It has been doing it in the entire time. And the answer was hiding in the branches. Welcome to the Scholar's Fire. You're listening to the Scholar's Fire, the Wednesday deep dive from the Wiretap, the show where we take one idea, one thinker, one study, or one tradition, and crack it open using every tool we have: neuroscience, anthropology, evolutionary psychology, and the accumulated wisdom of civilizations that figured out how humans work long before we had functional magnetic resonance imaging, fMRI machines. I'm Wish, and today we are doing a study breakdown, a format where we take a single landmark paper, explain what the researchers actually did, why it matters, and what it means for you, the wired ape navigating a world your brain was not quite designed for. Today's study is the Francioni and colleagues paper from the Harnett lab at MIT, published in Nature on February 25th, 2026. And I'm going to be honest with you, this might be the most important neuroscience paper published in this decade. This is not a hyperbole. This paper bridges the single biggest gap between AI and biological intelligence. It answers the question, how does a biological brain teach itself? And here's what you will walk away with today. First, you will understand what the credit assignment problem is and why it matters for everything from catching a baseball to overcoming trauma. Second, you will understand what backpropagation is and why scientists thought the brain could not do it. Third, you will understand what dendrites are and why they're wildly more interesting than your biology teacher let on. Third, you will understand what dendrites are and why they are wildly more interesting than your biology teacher let on. Fourth, you will understand what this MIT team actually discovered and how they proved it. And fifth, and this is the scholar's fire signature, you will understand how this finding connects to something ancient wisdom traditions have been saying for centuries, that the deepest learning comes from feedback within. Let us light the fire. All right, before we talk about Francioni and colleagues found, we need to set the table because this paper does not make sense unless you understand three things: the credit assignment problem, backpropagation, and dendrites. So let's talk about credit assignment problem. Imagine you are learning to shoot a basketball. You throw, you miss, your brain receives a signal single piece of information that was wrong. But that was wrong is useless unless you know what specifically was wrong. Was it your elbow angle, your wrist flex, your foot position, the force from your shoulder? What was wrong? Your brain has to distribute blame or credit across all the muscles, joints, and neural pathways involved and figure out which ones need a tweak and which ones were fine. This is credit assignment. And a simple system, it's straightforward. If you have one neuron connected to one output and the output is wrong, well, that neuron is the culprit. Case closed. The human cortex is not a simple system. The human cortex is a deep, layered, hierarchical network where the output of one neuron feeds into the input of another, which feeds it, which feeds into another, which feeds into another, layers upon layers. The deeper you go into the network, the harder it becomes to trace a mistake back to its source. It's like trying to figure out which specific domino out of 10,000 fell at the wrong angle and caused the whole chain to veer left. That is the credit assignment problem, and it has been called the central problem of learning in complex systems. In the 90s, computer scientists, most famously David Dromel Hart, Jeffrey Hinton, and Roland Williams, popularized an algorithm that solved this problem in artificial neural networks (ANNs). They called it backpropagation of error. And here's a gist: an artificial network makes a prediction. It compares that prediction to the correct answer. It calculates an error, and then here is the magic. It sends that error signal backward through the network, layer by layer, calculating at each neuron a specific tailored correction. Not a generic "you messed up," a precise mathematically calculated gradient that tells each individual neuron, "you contributed this much to the error in this direction, so adjust your correction strengths by this amount." And that tailored neuron-specific error signal is what makes backpropagation vectorized. A scalar signal would be a single number, like a thermostat saying too hot. A vectorized signal is a whole set of individually addressed instructions, like sending each employee in a company their own personalized performance review simultaneously. Backpropagation turned AI from a curiosity into a revolution. It powers virtually everything in modern machine learning. And here's the problem. For decades, neuroscientists looked at backprop and said, the brain cannot do this. And they had very good reasons. So why scientists said the brain cannot do backprop? Three big objections. Objection 1, the weight transport problem. In standard backprop, the error signaling traveling backward through the network has to know the exact strength of every forward connection. The backward pathway has to be a perfect mirror of the forward pathway. In the brain, feedback connections are anatomically and physiologically different from the feed-forward connections. They do not have access to each other's weights. It's like asking you to reverse engineer a recipe by tasting the final dish without ever seeing the ingredient list. Objection 2, the timing problem. In standard backprop, the forward pass and the backward pass happen in separate alternating phases. The network processes the input, then pauses everything to propagate the error backward. The brain does not pause. It processes feed forward sensory information and feedback error information simultaneously, in real time, while you're busy living your life. There's no hold on, let me update my weights break between seeing a ball coming at your face and talking. And objection number 3 is the locality problem. Backdrop requires each neurons to receive a signal that encodes global information about the network's overall error. How does a single neuron buried in layer 4 of the visual cortex know about a mistake that happened at the output layer? In artificial network, this is trivial. It's just math propagated through the code. In biological tissue, it requires an actual physical mechanism to deliver that information. For this reason, the dominant assumption since the 1980s has been the brain uses something else. Maybe something simpler, maybe something approximate, but definitely not real backpropagation. But in the 2010s and early 20s, a wave of theoretical papers from researchers including Blake Richards, Jordan Gergoyev, Alexander Pio, Richard Nord, Ray Pontecosta, and others proposed something radical. What if the brain's solution to the timing problem was not temporal separation but spatial separation? What if individual neurons could process feed-forward information and feedback error information at the same time in different parts of the cell? And they had a candidate for the hardware, dendrites. Quick anatomy refresher. A neuron is not just a little dot that fires. A neuron has an elaborate tree-like structure. The cell body called the soma is a central hub where signals are integrated and decision to fire is made. Extending from the soma is the axon, the long cable that sends signals out to other neurons. But reaching upward and outward from the soma are the dendrites, from the Greek word dendron meaning tree. Dendrites are the branching antenna-like structures where the neuron receives the vast majority of its incoming signals. Now, here's what makes this interesting. In the cortex pyramidal neurons, the major excitatory workhorses of the cerebral cortex have dendrites that are physically very long. The distal apical dendrites, the ones at the tippy top of the tree, furthest from the soma, can be hundreds of micrometers away from the cell body, and they receive different inputs than soma does. The soma and the nearby basal dendrites receive feed-forward information, sensory data from the outside world coming up through the cortical hierarchy. But the distal apical dendrites, way up in layer 1 of the cortex, receive feedback information. Top-down signals from the higher brain areas. Context, predictions, error information. This is not speculation. The anatomical wiring is well established. Feed-forward inputs arrive at the bottom of the neuron. The feedback inputs arrive at the top. The neuron, structurally, is already built to keep these two streams of information semi-independent. The theoretical model said, if dendrites can compute their own signals semi-independently from the soma, then individual neurons could potentially solve backpropagation like learning in a single phase. No need to pause, no need for symmetric weights, no need for global error broadcast. The solution would be local, spatial, and biologically plausible. Gorgeous theory, completely untested until this paper. Let's talk about the study design now. So Mark Harnett's lab at MIT designed what I think is one of the most elegant neuroscience experiments of the decade. They trained mice to perform a neurofeedback brain-computer interface PCI task. And here's how it worked. They used two-photon calcium imaging, a technique that lets you see individual neurons light up in a living brain, to monitor a small group of layer 5 pyramidal neurons in the retrospleen cortex of mice, RSC. They picked two tiny populations, 4 or 5 neurons designated P positive and 4 or 5 neurons designated P negative. The mice had to learn to control a visual grating, a pattern of lines on a screen, by differentially modulating the activity of these two populations. When the P positive neurons fired more and P negative neurons fired less, the grating rotated toward a target orientation and the mouse got a reward. The key design feature, the experimenter defined the reward function. They knew exactly which neurons were supposed to increase their activity and which were supposed to decrease it. This means they could calculate for every moment of every trial exactly what the error was, and then ask, do the dendrites encode that error? Critically, they did not just image the somas of these neurons. They simultaneously imaged these distal apical dendrites, the faraway treetops, in a separate imaging plane. Soma and dendrite, same neuron at the same time. This is technically ferociously difficult. Most neuroscience experiments image one or the other. Harnett's team did both. Alright, here's where it gets, and I'm using a technical term here, absolutely wild. Finding 1. Dendrites encode task-relevant information independently. The team developed a metric they call the somatodendritic residual, or ST residual for short. This is the difference between what you would predict the dendritic signal to be, based on the somatic signal and what the dendritic signal actually is. Think of it as this surprise factor in the dendrite. If the dendrite is just passively echoing whatever the soma does, the ST residual should be 0. Boring, predictable, nothing happening upstairs. But the ST residual was not 0, not even close. The dendrites were carrying information that could not be predicted from the soma alone. And when the team looked at what that extra information encoded, they found it tracked task-relevant variables, reward and error. The dendrites knew whether the mouse was making a mistake and how big that mistake was. They also confirmed that this dendritic signal was genuinely top-down by anesthetizing. They also confirmed that this dendritic signal was genuinely top-down by anesthetizing a separate group of mice. Under anesthesia, top-down cortical feedback shuts down, and sure enough, the ST residual collapsed. The surprise in the dendrites disappeared, and this confirmed that the independent dendritic computation required active, awake, behaving brains with intact feedback circuitry. Finding number 2, the signals were vectorized, neuron-specific. This is a big one. The word that changes everything, vectorized. It was not the case that all dendrites received the same generic error signal. Some kind of global things are going well or things are going badly broadcast. No. The error information the dendrites depended on the causal role of each individual neuron in the task. P-positive neurons, the ones that were supposed to increase their activity, showed dendritic amplification during error reduction epochs when things were getting better, and dendritic attenuation during error increase epochs when things were getting worse. P-negative neurons showed the opposite pattern. Read that again. Each neuron's dendrites received an error signal that was specific to that neuron's role in the circuit. The dendrite of a P-positive neuron did not get the same feedback as the dendrite of a P-negative neuron sitting right next to it. The signal was personalized, individually addressed. The brain was sending each neuron its own performance review. This is vectorization. That is the hallmark of backpropagation algorithm in artificial neural networks. And this paper demonstrated it happening in biological tissue, in living, behaving animals for the first time. Finding 3, dendritic signals predicted learning. The team found that the signs of these dendritic teaching signals, whether a neuron's dendrite was being amplified or attenuated, predicted how that neuron's activity changed over the course of learning. Neurons that received the correct dendritic signal early in training adjusted their firing rates in the right direction over subsequent days. In other words, the dendritic signal was not just correlated with error, it was predictive of the future learning. It was an instructive signal, a teaching signal, not just. Finding number 4, NDNF positive interneurons control the learning signal. And here's the causal proof, the experiment that closes the loop. The team used optogenetics, a technique where you can use light to control genetically specific neurons, to activate a type of inhibitory interneuron called NDNF positive interneurons. NDNF stands for neuron-derived neurotrophic factor. NDNF positive neurons live in layer 1 of the cortex, the same layer where the distal apical dendrites receive their feedback inputs. These interneurons are beautifully positioned to act as the gatekeepers of the top-down information. They have been described in the literature memorably as Spartans of the cortical column, small in number but massive in impact. They inhibit the apical dendrites of pyramidal neurons, controlling the flow of feedback signals. When Harnett's team activated NDNF positive interneurons with light during the learning task, 3 things happened. 1, reward-related information in the apical dendrites of layer 5 neurons was disrupted. 2, error-related information and signal vectorization in the dendrites was abolished. And 3, the knockout punch, learning itself was impaired. The mice could not learn the task. Let me say that clearly. When you turn off the dendritic teaching signal, learning stops. The signal is not decorative. It is not epiphenomenal, which is the scientist's polite way of saying just along for the ride. The signal is causally necessary for learning. The brain is running a vectorized credit assignment algorithm implemented through spatial segregation in dendrites, and NDNF positive interneurons are the gatekeepers of that system. What this means for the field, let me put this in context. For 40 years, the neuroscience community has operated under the assumption that whatever the brain does, it is not backpropagation. The brain must use something simpler, something approximate, something fundamentally different. This paper does not prove the brain does exact propagation. The researchers are careful about this. They use the phrase backpropagation like in the and vectorized instructive signals in the final Nature publication. They note that future work is needed to determine whether the error signals are calculated locally at each layer or transmitted across layers in the classical backprop formulation. But what they have proven is that the core computational principle, vectorized neuron-specific error signals that are causally necessary for learning, exist in biological neural circuits. The brain has its own implementation using the spatial architecture of the dendrites rather than the temporal phases of artificial networks. It's not a copy of backprop. it is arguably more elegant. It is a single phase solution that artificially intelligence engineers are not trying to replicate. The irony is staggering. AI was inspired by the brain. Then AI invented backprop. Then neuroscientists said the brain cannot do backprop. And now the brain turns out to be doing something that may be better than backprop. And AI researchers are scrambling to learn from it. We built the student. Now the student is learning from the teacher it did not know it had. Let's do the synthesis now. Let's see how ancient wisdom meets dendritic computation. Alright, if you have been a Scholars Fire listener for any length of time, you know this is where we do something no other neuroscience podcast does. We take the finding and we zoom all the way out. Because the wide depth thesis is not just that science is interesting. It is that science, anthropology, and ancient wisdom are often describing the same animal from different angles. So let me ask a question that might sound strange after 40 minutes of dendrites and optogenetics. What if contemplative traditions have been pointing at this mechanism for thousands of years? The central finding of this paper is that the brain teaches itself through internal signals, not through external reward alone, not just through a pellet of food or a pat on the back, but through internal neuron-specific error correction signals that flow from higher brain areas back down to the dendrites. The brain is literally providing feedback to itself from within at the cellular level. Now consider what contemplative traditions across cultures, across centuries have consistently emphasized. The Buddhist concept of vipassana, which translates roughly as insight through observation, is built on the premise that the deepest form of learning comes not from external instruction but from turning attention inward, observing your own body, your own sensations, your own mental processes, and allowing that internal observation to produce correction insight. The Vedantic tradition of Swadhyaya, self-study, similarly emphasizes that transformation arises when the system examines itself. The Stoic practice of proshke, attentive self-monitoring, rests on the same principle. These traditions did not use the language of vectorized credit assignment. They did not talk about somatodendritic residuals, but they articulated centuries before we had two-photon microscopes, a core insight that this paper now validates at the biological level. The organism learns best when it attends to its own internal error signals. Now let's talk about interoception and the dendritic feedback layer. Here's where it gets practically interesting and where we build the bridge to Friday's Ritual Friday episode. Interoception is a scientific term for the sense of the internal state of your body. Heart rate, breathing, gut feelings, muscle tension, temperature, the ongoing stream of information about what is happening inside you. And interoceptive awareness, your conscious ability to notice and attend to those signals, is one of the most trainable capacities in the human nervous system. Practices like the body scan, the systematic slow movement of attention through each part of your body, and somatic experiencing, a therapeutic approach developed by Peter Levine that focuses on bodily sensation, are fundamentally exercises in tuning attention to internal feedback. Now this study tells us that the brain's learning machinery depends on a specific internal feedback channel, the distal apical dendrites of the cortical pyramidal neurons. These dendrites receive top-down signals, predictions, errors, corrections from higher cortical areas. And the integrity of this channel is gated by NDNF positive interneurons, which are sensitive to the animal's arousal state and behavioral context. Previous research has shown that NDNF interneurons in layer 1 gain modulate whole cortical columns depending on behavioral state. They ramp up during alert, attentive states and modulate the balance between bottom-up sensory processing and top-down feedback. And here's where the wire-dip connection happens. Practices that cultivate alert, internally attentive states, focused but calm, present but reflective, may be creating precisely the neurophysiological conditions under which dendritic feedback signals operate most effectively. And when you sit in meditation and gently direct sustained attention to bodily sensations, you're not just relaxing. You may be optimizing the very dendritic feedback layer that this MIT study shows is essential for learning. You are, in a very real sense, training your brain to listen to its own teaching signals. So this is not a single culture phenomenon. The emphasis on internal feedback as a learning mechanism appears everywhere. In the Japanese martial arts tradition of Keiko, repetitive practices with deep internal attention, the student is taught to feel the correction from within, not to wait for the teacher's verbal instruction. The Sufi tradition of muraqabah, self-observation and watchfulness, is structured around sustaining attention to inner experience as the primary mechanism of spiritual refinement. Indigenous Australian Dardiri, deep contemplative listening, explicitly framed attentive internal stillness as a way in which understanding arises. So every one of these traditions says, in a sense, the system that learns is a system that listens to itself. And dendrites, it turns out, are the biological infrastructure for exactly that kind of self-listening. Now, I would not be doing my job on the Scholar's Fire if I just handed you the exciting version and left out the caveats. This is a landmark paper, but it's not the last word. Let us talk about what we do not know yet and where the debate is headed. Caveat 1. Mice are not humans. This study was conducted in mouse retrosplenial cortex using a brain-computer interface task. The retrosplenial cortex in mice is involved in spatial navigation and memory. And while it is a reasonable model system, it is not human prefrontal cortex during abstract learning. Whether vectorized credit assignment via dendrite scales to the complexity of human cortical computation, 32 cortical areas, billions of neurons, the full glory of language and abstraction, remains an open question. Harnett's team acknowledges this. The paper explicitly calls for future work examining whether error signals are calculated locally at each layer or transmitted across hierarchical layers in the way classical backpropagation requires. Caveat 2. Correlation versus causation, mostly resolved. A persistent concern in neuroscience is that observed signals must be correlational, just riding along rather than genuinely instructive. This paper does an unusually strong job of addressing this concern through the NDNF positive optogenetic perturbation experiment. But when you disrupt the signal and learning breaks, that is causal evidence. But the authors note that they cannot completely rule out that dendritic activity reflects moment to moment computational signals rather than teaching signals for the synaptic weight changes specifically. The interpretation is well supported but not airtight. Caveat number 3, the source of the signal. The paper demonstrates that vectorized instructive signals arrive at the distal apical dendrites, but it does not yet determine where those signals come from. Are they generated by glutamatergic inputs from higher order cortical areas, neuromodulatory signals, products of local recurrent excitatory and inhibitory computation? All three are plausible and each would have different implications for how the brain's learning algorithm actually works. This is the next frontier. Caveat 4, the wisdom tradition connection is an inference. I want to be transparent here. The link between this study and the contemplative practice is an inference, a plausible, exciting inference supported by converging evidence from interoception research, attention neuroscience, and the known role of NDNF positive interneurons in a state-dependent processing. But nobody has directly tested whether, say, experienced meditators show enhanced dendritic vectorization during the learning task. That study has not been done. When it is done, and I believe it will be, potentially within the ritual operating system ROS research framework, it could be transformative. But for now, it's a bridge, not a proven road. So let me tell you the wired ape perspective here. Where we come down on this as a wired ape is that this paper provides the strongest biological evidence to date that the brain implements a sophisticated vectorized internally generated learning algorithm at the subcellular level. It does not prove that the brain does exact backpropagation, but it demolishes the 40-year assumption that the brain cannot do anything resembling it. The biological solution, spatial segregation dendrites gated by NDNF positive interneurons, is arguably more elegant than the artificial one. And its resonance with contemplative traditions that emphasize internal feedback as a primary mechanism of learning is too strong to ignore. We're not saying meditation is backprop. We are saying that the human nervous system has a built-in self-teaching mechanism, and every culture on Earth seems to have independently stumbled onto practices that cultivate it. So let's talk about the ember. Here's your ember for the week. One idea to carry with you: your brain does not learn the way we thought it did. It does not wait for a gold star or a slap on the wrist. It does not need a teacher standing over its shoulder. Deep in the branches of your cortical neurons, in the dendrites, those beautiful tree-like structures reaching towards the surface of your brain, there is a feedback system that sends individually addressed correction signals to each cell. Your brain is teaching itself from within at a level of specificity that we have been building machines to approximate for 40 years. And here's the part that matters for how you live your life. The system is gated by your state. It works when you're alert. It works when you're attentive. It works when you turn your awareness inward. Every culture that developed practices of internal attention, body scanning, meditative observation, somatic awareness, contemplative listening, may happen without knowing the mechanism, cultivating the optimal conditions for this dendritic feedback system to do its work. You're not broken, you're not failing to learn. You might just need to listen to the branches. And this is your bridge to Friday, when we will walk through a specific practice, the body scan, with a new lens. You're not just relaxing, you're training your brain to hear its own teaching signals. If this episode lit something in you, subscribe to the Wired Ape wherever you listen. Follow us at the Wired Ape on Instagram, X, and all the usual suspects. Visit thewiredape.com for deep canopy article, reading list, and the episode resources. Check out cywester.com for the behavioral finance side of the Wired Ape brain and the management messiah on LinkedIn for how these insights play out in the workplace. Join the watering hole for community discussions, your questions, your insights, your expectations, and experiences. This has been the Scholar's Fire. I'm Vish. Understand the Wired Ape brain, work with it, not against it. We'll see you Friday.